Hi, my name is Caroline Kumsta, and I'm very excited to tell you today about the research that we are doing in my lab. I wanted to start with a question. What is the main risk factor for the development of these diseases? Diseases like cancer, heart disease, neurodegeneration, diabetes, or musculoskeletal diseases. It is aging. Age is the main risk factor for appearance of diseases. As you can see here on this graph, the incidence for these diseases really starts rising after the age of 70. And my research is really focused on figuring out why that is happening and how we can stop it. Particularly, I'm interested in understanding and preventing the gap between health span and lifespan. As you can see here, humans are spending the last 10, 15 years of their lives in ill health. And my goal is to reduce this gap. To do this, we're trying to understand what are the processes that lead to aging and trying to prevent them or even reverse them from happening. So, what is aging? Aging is really the slow accumulation of damage. And as you can see here, illustrated with this penny, you can see that this penny over time accumulates a lot of grime and dirt and doesn't look as shiny for when it was new. And this is all due to environmental factors, right? So this also happens in our cells. When we are young, our cells work perfectly. But then over time, through environmental, physical, chemical, and biological, so in cell intrinsic processes, we accumulate a lot of waste and damaged products. So what if we could stop this from happening? Or what if we could take this waste and actually eliminate it? And this is a cellular process in our bodies, in all of our cells, that actually does this. It's a cellular recycling process. It's called autophagy. And it does what just real life recycling out in the real world is doing. It takes all of the waste products that accumulate and recycles them down to their building blocks so that we can reuse them. My lab, we study aging and the cellular recycling process autophagy in tiny roundworms called C. elegans. They're really tiny, just a millimeter big, which is just like a comma on a page. And they're a great model organism to study aging because you can actually really see the aging process here. In young worms, these worms move perfectly and they look just like you see here on this picture. But when they're old, they become a little bit more sluggish. They accumulate, they become a little bit wider around their middle. They get more wrinkly. And this is how we can study these aging processes. So what we have found using these worms and the cellular recycling process, we have found that with age, the recycling process declines with age. We see that there's reduced activation of the cellular recycling. We also see that the waste transport is slowing down. So basically the recycling trucks are not moving as efficiently and quickly any longer. And then also there's inefficient elimination of the waste. Basically the cellular recycling factories stop working. So what we're trying to figure out is how can we increase and boost this process? And what we have found is that if we activate this process, this cellular recycling process early in life when these animals are young, we can actually extend their lifespan. As you can see here from the survival curve, the ones with the activated autophagy processes in blue live longer than the control group in gray. So how do we activate this process of autophagy? We do this with mild heat stress. Think sauna. We do this also through dietary restriction. Think intermittent fasting. and we can also do this via exercise. And yes, we can exercise the worms. So how do we bridge this gap from worms to humans? So what we wanted to figure out is, does autophagy also decline with age in humans? But until now, it was really difficult to measure this process in human cells. So now there is a method where we can use just blood cells, so a simple blood draw, to measure the age-related effects of this process. We can also take skin cells from donors that are either young or old, and we can measure this process. And then we can also make different human cell types out of the skin cells to measure and see what is happening in, for instance, neurons that we're making out of these. And what we have found is that in blood cells, there are differences between young and old humans and autophagy. Next, now that we can measure this process, we can test whether interventions restore autophagy to more youthful levels. Again, fasting, sauna, and exercise. And we have started doing the clinical trials with our partners at UCSD, where we let people undergo a mild strength and stability exercise program for 12 weeks. And we measured their autophagy status in blood before and after. And we could show that this exercise program restored autophagy levels to more youthful levels. So with this, my research from worms to humans, what we do is we measure cellular recycling, we restore it safely to promote healthy aging. Thank you.